permanent compound tissue. So first, in permanent compound tissue there are two things, xylem and phloem. Okay, so what is xylem? First we will discuss xylem. We know xylem and phloem are, why they are compound? Because they are made up of different types of cells, right? Different types of cells are present. The elements of xylems are known as xylem elements. Okay, first you see there are four types of cells. The cells, these dead cells are vessels or tracheids. Vessels we will call it in angiosperm, tracheids we will call it for gymnosperm. Dead cells is xylem fiber, that is sclerenchyma, and xylem parenchyma is the living part, right? So now we will draw the structure of vessels and tracheids. So vessel is for angiosperm, we will call it vessels. Tracheids for gymnosperm. You see the structure of vessels. Vessels are some tube-like structure. The one above the another, one cell above its another cell. In this way they are arranged. But you see there are here is some side by side cells are also there, right? Between two cells there is a pit, means the connection between these two cells we call the pit, and these are the lumen, and here is the lignin deposition, right? That's why they are dead cell. Same for tracheids. In tracheids this arrangement is like this. So these are the connections. Okay, this is the pit. So here also how the water will flow in this direction, sidewise then upwards, sideways then upwards. Here's upwards, sideways, upwards, right? So if there are turns and bends, you see there are turns and bends in the path of anything, the flowing rate of conduction is not going to be that much efficient, right? Here you see there is a easier way, but here this is like this, so there is a more turns, right? So that's why it is same. Okay, so it is not going to be efficient. That is why the vessels are more evolved type of tissue compound to tracheids. So vessels are more evolved. That's why angiosperm, this is found in angiosperm. So angiosperms are most evolved type of plant than gymnosperm. Clear? So if we draw the inner part of this structure, a section under microscope, if we see, we will see the lumen is so small because in the wall the more lignin deposition is there. So that's why the more lignin deposition in the wall, that's why the lumen is small. But here in tracheids, in the wall the lignin deposition is less, that's why the lumen is more thicker, right? So this is the difference. So if we see a xylem tissue under a microscope, how we will identify which cell, what are the xylem elements? So you see these are the cells. You see there is a pit, that means it may be tracheid, it may be xylem, it may be tracheid, it may be xylem, right? Around which you see some cells are present, right? These are the xylem fiber, and these are the xylem parenchyma. Clear? So xylem parenchyma, we know the function of xylem parenchyma, it stores food. Xylem fiber gives strength. Xylem vessels, and vessels means tracheid, and trachea helps in flowing of water, right? Helps in flow of water, right? Conduction of water. So there is two terms we will learn about xylem. That is, one is the protoxylem and metaxylem. Protoxylem is the first from xylem. Okay, where in protoxylem the xylem vessels are small, but in metaxylem, later formed xylem, is at the metaxylem where the xylem vessels are larger. So we know the function of xylem is conduction of water. Now we will learn about the phloem. What are phloem? We know phloem elements. First we will learn sieve tubes, sieve shells. Sieve tubes is for angiosperm, sieve shells are for gymnosperm. Companion cells, or we can say albuminous cells. So you see these two cells we could call sister cells because they form from one parent cell. Okay, and xylem fiber also we call burst cell. So phloem not xylem, phloem fiber, phloem parenchyma, in which only xylem fiber is dead, other all cells are living, right? Achha. So you know the sieve tube or sieve cells and mature sieve tubes and sieve lack of nucleus. Okay, there is nucleus is not present. If I think you know that give one example of cells where nucleus is absent in plant cell, then we will give the example of sieve tube, because in which plant the nucleus is absent. Achha, so if we draw the sieve tube and sieve cell, how will be the structure? You see there are some perforated sieve. These are the lumen or tube you can see, and these are the sieve plate. Okay, through this sieve plate the food can pass. And sieve shells, here this perforation is only in different places, but you see in the perforations are arranged in a particular area. Okay, these are called the sheath areas. So here the conduction will be more better than this. Okay, acha, they are called sister cells because they are formed from one parent cell. And you see, if it is shifted of shift tube, along with this the companion cell is attached, and companion cell have nucleus. Clear? Now you see, if you see the phloem under microscope, what we will see, you see these are if the sheath tube, so this is the phloem tissue. If the sheath tube is arranged in this way, sheath plate is present, then this will be this, and here the companion cells are attached. These are the burst cells, mean phloem fiber, and these are the phloem parenchyma. Same thing will be for this. If the arrangement is like this, then there is the sheath areas, companion cell, xylem parenchyma, burst cells means sheath fiber. Okay, so we know xylem and phloem together known as conducting tissue or vascular tissue. Similar terms for protozylem, protophlloem, metaphlloem. Protophlloem means the first form phloem, metaphlloem is the later form phloem. Clear? Now the tissue system, how this xylem and phloem are arranged. You see, first when all the cells are arranged in a plant tissue, the first one will be the epidermal cell. Next there will be a ground cell. In ground cell, parenchyma, collenchyma, sclerenchyma is present. Okay, then will be the vascular tissue system, that means xylem or it may be phloem. If we draw a structure, you see these are the phloem. And how will be the xylem and phloem arrangement, that I am telling you. You see, these are the xylem and this middle one are the phloem. Okay, so these are the radial, you see the radial wise these are arranged, right? So we will call this radial arrangement. We see this type of arrangement in roots. But if we see the xylem, phloem is on outer side, xylem in inner side, is a flower like arrangement. Phloem is outer, xylem is inner. This is known as conjoint arrangement. We see this type of arrangement in stem. We can classify this in two categories, collateral and bicollateral. If phloem is, this is for collateral. If you see, phloem is outside, xylem is inside. But if both the sides phloem is present and xylem is in middle, this is called bicollateral. So arrangement of xylem and phloem, we can classify congenital arrangement in these two ways. But if on the basis of presence of or absence of cambium, we can also classify this and congenital arrangement in two ways. If between xylem and phloem the cambium tissue is present, then we will call it open. Why it is open? Because secondary growth can take place because due to the presence of cambium tissue. Here you see phloem and xylem, in between there is no cambium tissue, so no secondary growth will take place. So this we see in monocot stem, this we see in dicot stem. Next one, that is the centric vascular bundle. Centric vascular bundle we can classify in amphicribal and amphivessel. In amphicribal, what we see, you see xylem is in the middle and phloem is circulating in outside. Here is the opposite thing, phloem is in the middle, xylem is in outside. So in the basis of this is also a type of vascular bundle arrangement. Okay, means xylem phloem arrangement. Now this is a more important, exerg and enderg. Exerg and enderg condition means arrangement. We know protoxylem means the first form xylem where smaller vessels is present. Metaxylem is later form xylem where the xylem vessels is larger. So what will be the arrangement for exerg condition? You see, exerg condition we see for mostly we see in roots. Okay, so you see this is the outer side. This is the outer side. These are the meristematic tissue, and we know meristematic tissue give rise to new tissue. Below the meristematic tissue, protoxylem is present, and meristematic tissue will give rise to new tissue. If the new tissue arranged below this protoxylem, okay, and next there will be again a new tissue will form, and this new tissue you see again this below this structure, right? So first protoxylem ওর নিচে হচ্ছে নিউ টিস্যুটা যেটা ফর্ম হচ্ছে মেটাজাইলেম, তারও পরে মেটাজাইলেমটা যখন ডিভাইড হয়ে আরও বড় হচ্ছে সেটার নিচে অ্যারেঞ্জমেন্টটা হচ্ছে। সো ইউ সি protoxylem is outside, metaxylem is inside. This type of arrangement we call exerg condition, where protoxylem is outer and metaxylem is inner. If the opposite thing happen, you see this is the meristematic tissue, these are the this is the protoxylem. If the new cell that is formed, this arrange above the protoxylem, এটার ওপর এটা অ্যারেঞ্জ হয়ে গেছে, দেখো এটা তাই নিচে শিফট হয়ে গেছে, আবার যখন নিউ সেলটা আসবে তখন এটা এখানে চলে আসবে, এর উপরে নতুন সেলটা আসবে। সো ইউ সি ইন দিস ওয়ে প্রোটোজাইলেমটা কিন্তু নিচের দিকে চলে যাচ্ছে, নিউ সেলগুলো ওপরের দিকে অ্যারেঞ্জমেন্ট হয়েছে, ঠিক এটার অপোজিট। সো এটাকে আমরা কি বলবো, এন্ডার্স কন্ডিশন, ওয়্যার প্রোটোজাইলেম ইজ ইনার অ্যান্ড মেটাজাইলেম ইজ আউটার। দিস ইজ আ ক্যারেক্টারিস্টিক ফিচার অফ স্টেম। ওকে।